This tutorial outlines bulk RNA-seq analysis in the Europa Data Explorer. First, we upload files. Files are uploaded in the upper right from a number of sources, including locally from your computer, AWS storage, or external sources such as Geo. The Geo search feature allows us to use human-readable text or an accession number to find studies of interest. Here, I'm looking for studies associated with rhinovirus, dendritic cells, and anti-IL-5 therapy. The agent finds studies, such as this one, gives some basic information including the original title and a summary or abstract. We can also see the full Geo submission with the Details button. Directly from this search screen, we import the data into Europa. Here, we continue to see the information about the study as well as details on each file. We can include individual FASTQs using the toggles or the Upload All button. We can also upload the already aligned counts table if the authors included one in Geo. I previously uploaded these files, so I'm just going to close out. Once the files are uploaded, we see a report for each FASTQ from FastQC. This report is run automatically on all FASTQs uploaded from any source. Here we take note specific parameters such as adapter contamination in this plot, as well as duplication levels and potentially overrepresented sequences like primers in later plots. We can easily toggle between files and use the links to look at the specific plots of interest. In general, at this stage, we're determining parameters that go into the next step of sample creation. We can also see the Geo matrix, which is simply a CSV of counts per sample per gene. We visualize this with a heat map, which gives an overview of the data. Here, you may see poor quality files as outlying columns, but the data is so rich that it's difficult to see patterns at this stage. To create samples from FASTQ files, we select one or more FASTQs. Then we create samples and provide the parameters for quality filtering and alignment. These samples are from human data, and from the FastQC reports, we saw that there's about 15 base pairs of adapter contamination at the start of sequences. The remaining defaults work for these data and follow the original publication. Note that there are 3 possible aligners, though we're using the default star. Additionally, if there are adapter or primer sequences found by FastQC or custom ones that we know are there, they can be trimmed here. For these data, these sequences do not exist, so these parameters are not needed. As samples are being created, we can see their progress in the upper right. Here we see that we've initiated our sample creation, and under processing, we can see that 42 samples are in progress. Once they're complete, as we see here, the logs populated, and if there were any errors, this is where we could see where problems had arisen. Once samples are created, we move to the Samples tab. Here we see all of our samples visualized by expression on a linear or log scale. These are quick overviews to look for outlying samples or errors. To analyze these samples, we select them and create a study. Here, it's a good idea to add tags so that we can quickly sort through data in the future. We could have also added tags to files and samples prior to this. I'm going to name this by the GeoID so that I know what I'm working with. Because this dataset contains 2 experiments, the similarity index, meaning how many genes are found across all samples, is not 100%. This is expected since the 2 datasets were filtered separately in the original study. Optionally, we can filter to only genes found across all samples, which, as we saw in the similarity index, removes less than 0.2% of genes. Once the study creation is initiated, it will appear in our Studies tab. While running, we'll see our running icon. Once complete, we can open the study using the Erlenmeyer Flask. The default box plots will show us the overall expression per sample. We can toggle on and off the outlying dots to view individual genes within samples. Here, some samples appear to be low quality and have very few counts. That's even more apparent when we turn off outliers and see that the medians are basically 0. This is expected as the original study details some failed samples are present in the data. Next, we're going to recapitulate the analyses from the original study. The first thing to note is that they analyzed their 2 experiments separately, but included both experiments in a single geo submission. In order to differentiate experiments, we need to annotate our samples. In the Manage Annotations tab, there are a number of ways to add this information. We can simply use the text editor to input information into a column. And if we need to add additional columns, we use the plus symbol on the right. However, if we have a lot of annotations, this may be slow. Instead, we download the sample names and then upload a completed table matching these names. Here, I downloaded the sample names from Europa. Then, in a separate file, I completed the annotations matching these names to the information found in the supplemental data of the original manuscript. For example, we have library IDs, pass filter, sample source, etc. We then upload this file into our annotations manager to add it to the Europa study. The annotation file doesn't have to be perfect before we upload because the annotation manager allows us to make further changes. First, we identify which column in our current annotation matches a column in our complete annotation. Here, they're both called name, but they don't have to be. We see that 42 samples are successfully matched. We then select variables to include in the upload, adding or removing individual ones as needed. Once uploaded, we can continue to edit the annotations by hand if needed. Importantly, we check the data types to ensure the correct assignment of group versus numeric data. For this study, we only have group annotations. However, we may have both types or may need to modify a group annotation that was incorrectly assigned if we had numeric data. Once saved, we can now sort and group our data based on any of the annotation. Remembering that there were 2 studies here, I'm going to break this up by experiment as well as pass filter. We see 3 groups, including the pass filter samples for the 2 experiments, as well as failed samples for one of the experiments. Deselecting all and then selecting just the failed samples, we now note that these are the ones we saw in the box plots with very low expression. So they will not be included in analysis similar to what was detailed in the original study. Next, we select samples that pass filter in the first experiment. To subset to these data, we go to our Dataset Actions menu under the plus sign. This menu includes all actions that modify the dataset, including filtering, normalization, statistics, and merging. We will use a number of these steps throughout this tutorial. We select Data Filtering by Selection and give the new data subset a name and a description. We see here that we are selecting 15 of the samples and all of the genes from the original data. Next, we do the same steps for this larger second study. Again, we go to the plus symbol, filtering by selection, and provide a name and description. This subset has 21 of the 42 samples. Let's look at the first study. Here we see our subset of samples and the box plots for their expression. Let's again group by annotations of interest. Looking at the IL-5 and viral annotations, we see that these samples are related to eosinophils and rhinovirus. So let's go ahead and change this dataset's name to be more descriptive, including EOS for eosinophil and RV for rhinovirus. Similarly, we rename the other subset something more specific to that experiment. Let's now review data cleaning in the original publication. We see that their data cleaning included protein coding genes, TMM normalization, and filtering of rare genes at 1 count and at least 3 samples. We can perform these same steps in Europa. First, let's filter for protein coding. We go back to our Dataset Actions menu and select Data Filtering by Biotype. By default, protein coding will be selected, but you can select one or more of the other biotypes as you need. Here, we also want to be sure that we've selected the correct column for our unique gene IDs. By default, if data were processed in Europa, the IDs are ensemble, but you may have HGNC or MGI symbols or other identifiers. The new dataset appears in our tree. Once complete, we note that originally we had 63,000 plus genes, and after protein coding filters, we have just over 20,000, which is an expected amount for a human RNA-seq. Next, we're going to filter by expression to remove rare and low abundance genes. Here, you can add a description and tags if you desire. The defaults match the original publication with 1 count and at least 3 samples. However, if you have a specific group of interest, you can subset within group using the second option, samples in at least one group. This can be useful if you want to make sure that your genes are present in, for example, both control and infected samples. Looking at our abundance filtered data, you can see that the scale and look of the box plots significantly changed as we removed many of the zeros. Here, our medians are above 0 and our box plots have both whiskers showing distributions. Let's move on to data normalization next. There are quite a few options here, including Z-score, median polish, percentile, etc. We select counts per million with TMM, which is the default for the EdgeR pipeline and what was used in the original publication. By default, this step automatically selects the sample closest to the median across all samples as a reference. However, if you have a particular, for example, control sample you want to force to be the reference, you can set that here. For this study, we're going to leave it as the default, and it will select the best sample to use as reference for the normalization. Once normalization is complete, we can select the dataset to see our cleaned data. Here we see that the scale of our box plot is significantly changed now that we're in counts per million. At this stage, we've employed steps in Europa to recapitulate all of the data cleaning done in the original publication. Next up, let's do some analyses of the data. All of the data cleaning steps were under the plus symbol because we were modifying the dataset. Now we're interested in analysis, which can be found there or in the column pane here. Clustering can be done with many different methods, and here I'm going to employ my standard, which is hierarchical clustering using Euclidean complete distances. We can cluster rows, which are genes, and/or columns, which are samples. We see that there are many metrics, and for each metric there may be multiple linkage types. There are some notes in the information icon about specific clustering methods. While this runs, we'll also perform principal component analysis, or PCA, as our next analysis step. The only parameter here is how many components we want. Here we leave the default 3. If you recall, we already annotated our samples. As a reminder, those can be seen from the upper right in the sample annotations. We can also add annotations to our genes. By default, we have our ensemble IDs, which are very specific to genes and genomic positions, but aren't very human readable. Using row annotations in Europa, we have the same setup as with columns, where we can add annotations, upload and download files, and change annotations in the manager. There are also pre-built annotations for genes that you may find useful. For example, here we have human protein coding annotations already uploaded in the system. Because we have ensemble IDs in this study, we can match to that column from the database and add information that we find of interest. For example, I'm going to add the genomic position using chromosome, start and end, as well as the gene symbol, which is the human readable names that you may be familiar with. There are some unmatched genes, because not every ensemble ID has a symbol, which is expected. Once complete, we save the changes. And because this file is quite large, we are prompted to refresh when it's complete. While we were doing this, we see that our 2 analyses completed. So let's take a look at the clustering first. We can use the full screen view in order to see this large heat map. There are a number of settings to change the look, including log transformation, and this is a dynamic HTML plot where we can hover to see information for each cell. We can also start to look at how our samples group in the clustering using column annotations. For example, we see that rhinovirus has a large impact on the transcriptome, with those samples tending to cluster together. In contrast, eosinophil treatment has less of a global impact. We can also force grouping by these variables outside of clustering with the group by function. And because we clustered by rows and columns, we can also turn on and off that clustering. Finally, we can change the colors to predefined palettes or to a specific set of values per color. Let's return to the quadrant view and look at our PCA. Similarly, viewing in full screen, we have a dynamic HTML plot that allows us to rotate along our 3 PC axes. If we calculated more than 3 PCs, we could change them here. We can color by our column annotations here. Let's color by rhinovirus since that was the larger effect in the heat map, and unsurprisingly, we see some nice splitting in the PCA as well. If we're interested in just 2 PCs at once, we can use the presets to view each side of the 3D object. We can also change the look of the plot with perspective lines, axes labels, legend, etc. Finally, this plot also auto-calculates the centroids for each colored group. And you can download the plot both locally as well as to the notebook within Europa. Next up, we're interested in statistical analyses of these data. Before we define any column sets, if we select statistical analyses, there aren't any groups to choose from. Under linear modeling, we could test individual variables with covariates. However, this does not give us the combined 4 group levels we're interested in. Thus, we need to group by those annotations in order to run a test. Here, we have our eosinophil and rhinovirus groups, which I'm going to order from bottom to top based on what I want as a reference. This is not required, but I find it helpful. We have our untreated controls, which we're going to rename with a shorter name, virally infected, eosinophil treated, and eosinophil plus virally infected samples. We can then save these into a column set. Selecting this column set, we see that we've saved our groupings, and these groupings can now be used in a statistical test. If we select our groupings, we see our levels. When we select only 2 levels, such as control and RV infected here, we see that we have a number of statistical options including t-tests, Mann-Whitney, and ANOVA. However, when we have more than 2 groups, we'll see the options change to an all by all, which means all pairwise comparisons between each of the groups. If we instead wanted to use a linear model, in this case, we go back to our original annotations, such as virus, and then perhaps use the IL group as a covariate. But in this case, to follow what the original publication did, we're going to use our grouping in an all-by-all t-test. Additionally, it's standard to log transform RNA-seq data before statistical tests due to the skewed nature of the counts. Because we're running across thousands of genes, we're also going to apply a false discovery rate correction using the Benjamini-Hochberg method. This again follows what was done in the original publication. We could run this test as is, or we can also add gene set enrichment analysis at this stage. To do this, we define what our gene identifiers are, verify that we have the correct species, and then pick a Broad Molecular Signatures database. Here we're going to use Hallmark. Additionally, we can filter pathways that have too few genes represented in a cleaned dataset. This is important for targeted panels and microarrays. However, for RNA-seq, we can set this to 0 since in theory we have measured every gene in the transcriptome. We then see our statistical test appear in our data tree. Once complete, we can visualize the results. The first plot that we see shows the significance for p-values and adjusted p-values in a histogram. We can change the look of the histogram by bins, as well as look at our different pairwise comparisons. Next, and probably the most popular plot for DEGs, is the volcano plot. Here we can see adjusted and unadjusted p-values with ratios or fold change. We have a note, because we log transformed our data, it recommends using the log2 ratios accordingly. If we click on a dot, we see the box plot of expression for that gene. Note that the gene's ratio is positive, but it looks negative in the box plot. This is just because of the ordering of the variables. We can switch those such that the positive values are upregulated by virus and the negative values are downregulated. We can also see the p-values for all of the pairwise comparisons in this box plot. GSEA results are contained in the biological context section. Here again, we see all of our pairwise comparisons, and we can view the results by selecting one or more. The first plot shows our normalized enrichment scores for each pathway. Again, remember that the ordering is opposite, so negative would be upregulated by rhinovirus. We can view the overall significance histogram similar to the gene-level statistical test. The results are also shown in several other visualizations, including the original dot plot, a bar plot, heat map, and a clustered heat map. The heat maps are particularly useful if you want to compare results from multiple pairwise comparisons, such as control versus our 3 treated groups, here. We see that in general, rhinovirus has a similar effect regardless of eosinophils being there, but eosinophils alone have a very different effect than rhinovirus. If we ran reactome pathways, we could also view that here. The final option is to view the results table, where we have a list of the leading edge genes and the other results. Here, or in any plot, we can select a gene set to see a highlighted volcano plot where the colored dots show the leading edge genes. Again, take note of the order of the selected groups so that the ratios make sense. Another type of pathway analysis you may be interested in is overrepresentation analysis, pathway enrichment, or Fisher's exacts test, all meaning the same thing. This is where we test if significant genes are overrepresented in pathways. Here, I happen to know that the negative log 10 of FDR.2 is about 0.7, so I'm going to select that cutoff as well as a ratio of 1. When I select the upregulated genes and save the list of genes as a gene set, we can then ask if those genes are overrepresented in any pathway. To do a comparison, let's also look at the effect of eosinophils on their own relative to virus. We employ similar cutoffs and select the upregulated genes again. These genes will appear as row sets in our row pane. When we select these, we'll see that the volcano plot is automatically filtered for just those genes, and we can change the comparison as needed. We can analyze these enrichments, or these for enrichment, using the Fisher's analysis tool. We select our gene IDs, species, and database to compare to. We run this for both of our sets separately. Once complete, we'll see it's ready, and the results will appear back in our biological context section under the Fisher's exact test, or FET. Similar to GSEA, we have a number of plots, such as dot plots, and selecting a gene set will show us the volcano plot highlighting the genes that were significant and in the selected pathway. Here we can also view and download the results table, including K/K enrichment, significance, and size of pathways. Similar to GSEA, we can compare the signal from these two gene sets using our clustered or unclustered heat maps. Another way we may wish to look at our different gene sets is by looking at how many genes overlap. We go back to our data set and use the Venn diagram option. We select sets to see which genes are present in only one significant group, or the other, or both. By selecting one or more of these, the hashing shows our selection and we get a subset of the gene sets. We can use this as a new set and do additional enrichment or visualization. Note that we can also visualize the reactome pathways here for the entire data set. Another additional analysis you may be interested in is correlation. Here, because we still have gene sets selected, we see only the expression of selected genes in two samples, or selecting different samples, or across all pairwise comparisons. For example, a sample compared to itself has perfect correlation seen here, and we can change our color range such that we see more subtle differences. Now we see a bit of a patchwork pattern, which, knowing our original sample order, is alternating rhinovirus infected and uninfected samples. If we instead go back to all genes, we see more variability in correlations between samples. Again, note the patchwork look since our infected and uninfected samples are alternating in our original sample order. Finally, let's consider the second experiment. Here we have our other data set using anti-IL-5 therapy. Using the guided analysis OWL, we can perform a standard workflow similar to our eosinophil dataset, including biotype filtering, rare gene filtering, normalization, and a simple statistical test. We only have our prior groups here, so we need to go back and create new anti-IL-5 groups for this second dataset. Again, we group our annotations of interest, order them, create a column set, and then we can use this column set in our statistical test. Note that the standard workflow applies a simple t-test. If we wanted to do more complex statistical tests or use other methods, we need to run that from the full menu as we did for the other dataset. However, guided analyses are a great way to get started, and you can save your own custom workflows to standardize methods across studies in future. Once complete, we will have all of the steps for the second data set that we had for the first, except for clustering, which isn't run by default.